Hi, my name is Pol Alonso. I am a postdoctoral scientist in the Institute of Material Science of Barcelona, belonging to the CSIC. And here, in the framework of the project NextGEM, we work with the model organism C.elegans in order to investigate the biological effects of electromagnetic fields. C.elegans is a very useful model organism because mainly two reasons. The first reason is that it has a very quick life cycle in which in just 3 days it goes through 4 larval stages and reaches the adult stage. And from here, the whole lifespan lasts approximately 2 or 3 weeks. This allows us to make very quick experiments assessing the effects of chemicals, radiation, and many other factors on the lifespan of the organism. Also, it has a very small size. The adult worm measures approximately 1 millimeter. This allows us also to have very large populations, a very large sample number, which makes the statistical power greater. Although C.elegans is very small, it has well-differentiated structures in the body that resemble the human body. For instance, the nervous system is clearly visible here in green. These are the neurons spanning the whole length of the worm. Also, the intestine is clearly distinguishable here in yellow. The worm also has a reproductive system, which is the blue part of the worm, this blue organ here. Also, the muscles are clearly distinguishable here in pink. And finally, the pharynx. The pharynx is the mouth of the worm, and it is a very interesting organ because it has a pumping movement which resembles the human heart. So, summarizing, although C.elegans is very small and one can think that it's very far apart from humans, it has clearly distinguishable structures in the body that allows us to make parallelisms with the human body and allow us to make studies which are extrapolable to humans. As I said before, humans and C.elegans share common features that allows us to make parallelisms between the two organisms. The brain in humans is represented by the ganglia located in the C.elegans head. The heart in humans is represented by the pharynx pump that I mentioned before, which pumps food, not blood, inside the gut, but nevertheless it has a structure and functioning which is very similar to the human heart. The gut is also another organ that is shared between the two organisms. The reproductive system also is here in C.elegans and allows us to make to model the reproductive system and development of humans. The muscles, and finally the neurons are also clearly distinguishable in C.elegans. The fact that a worm, which is a very easy to maintain organism, share these features with humans allows us to bridge the transition from in vitro experiments, which are cells on a plate, and in vivo experiments involving organisms like mice, fish, which are hard to maintain and experiments take longer. C.elegans, it's a good trade-off between these two stages, and it's the main strength of these organisms. We are now at the biolab, where the work with C.elegans is done. Here, Alejandro is synchronizing the C.elegans population in order to perform experiments. The bleaching is a method to synchronize the population of C.elegans. It's very easy. It's just treating the worms with bleach. So the bleach kills everything except the eggs. Then at the end of the treatment, you have a lot of eggs, and then you put these eggs on a plate, and the worms hatch at the same time. So you have a population of worms that all have the same age. Here we are looking at C. elegans individuals. These are adult worms, approximately 1 millimeter long, and these here, which are a bit smaller, are probably L4, the last larval stage. They are crawling on a lawn of bacteria, so you see these waves in the medium. It's a bacteria. Is the food of the worm. And here Julia is exposing the worm to nanoparticles to investigate the effect on the worm.